So it's been a real pleasure to visit Waterloo again. I was here when I was still at University of Toronto about 12 years ago, I think, and I've visited many times. I've been here for PhD defences back in the early 1990s in Richard Oakley's group, and we're now located in Bristol on the west side of England. And what I'd like to talk about today is this process which we've termed crystallization-driven self-assembly, or crystallization-driven living self-assembly. And it's an area that we kind of stumbled into. In fact, most of the areas that we work on in our group, they're really areas we kind of stumbled into and then sort of found them interesting and continued to develop them. And in our group, we sort of think about synthesis over different length scales. And when you think about making new compounds or materials, as many synthetic chemists do, usually you first meet this when you think about making molecules. And when you learn chemistry as an undergraduate, you invest a lot of time in courses where they show you how to make molecules. And this area is very well developed. I mean, it's remarkable the complexity that can be devised in molecular structures. So there's beautiful synthetic methods developed for making molecules. And there's still many very interesting challenges in this area as well. But certainly this area is very well developed. The achievements are really impressive. If you move up the length scale from molecules up to macromolecules, this is less developed, but in the last 10, 15 years there's been major advances here. Really important new synthetic methods for making polymers, which rely on methods called controlled radical polymerization, allow you to control the length of polymers and make very intricate structures. These are called living polymerization methods. And there's really a lot of advances here, but again, there's still many challenges. We work in both of these areas in our group. We have projects making molecules and also making macromolecules. But what I'd like to talk about in this lecture is making nanomaterials, which you can kind of define as materials that have length scales of between about 10 nanometers and a number of microns here. This is a field which is of enormous current interest, but really there's a real need for new synthetic methods for making structures in this size regime. And it's worth pointing out this is a size regime where biology is incredibly efficient. Biology has no problem making incredibly intricate, beautiful, functional structures such as viruses, proteins and so on within this size regime, and blood cells and so on. But synthetic chemists are really learning in this size regime here. And so we're interested in trying to find new synthetic methods that can be used for making structures that have a size of between about 10 nanometers and 10 or 100 microns. So the process that I'm going to talk about, and this is a method that a lot of people use to make nanomaterials in the size regime, is self-assembly. So the idea is you start with smaller structures, which are easier to make, and actually self-assemble them so they build into larger structures. So this is the method we're going to use, and it will involve block copolymers as the starting material for this process. So the basic aim here is, and the question we're trying to ask, can we develop new synthetic methods that instead of giving us molecules or macromolecules, but will actually allow us to make nanomaterials over this type of length scale? And what sort of desirable features do we want from synthetic methods? Again, you can look to biology as an example of what you'd like to achieve. You want to be able to control size. So if you want to make a structure that's 50 nanometers long, for example, you can do it. Or if you want to make 30 nanometers or 500 nanometers. You want to make structures that are monodisperse. So you don't want to have structures which have a broad range of lengths, for example. Biology, when it makes structures, they're all the same size. And so we want to make monodispersed structures, all the same size. We want to be able to spatially control chemistry and functionality. So if you make a structure, you might want to put different functionality at different points on on the structure. Again, biology is fantastic at doing this, but synthetic chemists so far have a real problem with doing this in this type of size regime. So these are some of the things that we sort of aim for in developing a method of this type. So the approach I'm going to talk about is block copolymer self-assembly, and we're going to start with block copolymers, which are basically segmented structures where you have two homopolymers joined together at a junction. And it's well known that these materials will self-assemble if you put them in a solvent that's good for one of the blocks but poor for the other, into core shell nanostructures or micelles. And most commonly, the morphology you get is a spherical structure. So if you take this block copolymer here, put it in a solvent that is poor for the blue block but good for the orange block, then you get a core of the blue block and a shell of the orange block. And these are colloidally stable structures in solution. So this has been very well developed where the two blocks are organic blocks, and in particular where the two blocks are amorphous. And what's known in virtually all cases where you take block copolymers of this type is they tend to give you spherical micelles. In some cases you can get other structures such as cylinders or vesicles. It's quite difficult to predict which structure you're going to get. Normally you get spheres, but if you want to make cylinders, it's very difficult to dial in the formation of cylinders into this block copolymer. You can play with the ratio of the two blocks, but often you just have to look at a wide range of materials and see what you get during the self-assembly process. It's very difficult to predict what you're going to get. And cylinders, for example, are a very interesting morphology. But when you can make them, you find that often they're actually mixed with other morphologies. So you'll make cylinders, but there'll also be some spheres or vesicles mixed in with them. So cylinders, for example, usually comprise very restricted range of the region of the phase diagram. The other thing to note about these structures is they're much more stable than micelles formed by molecular surfactants. So typically if you make a micelle out of a soap, for example, when you remove the solvent, they fall apart. These structures are much more thermodynamically and kinetically robust, and so you can actually remove the solvent and retain the structure. So you can think of actually doing interesting material science with these types of structures. And you can even, as I'll describe, actually make them permanent structures, fix in the structures so they won't disassemble. And this makes them, I think, very promising for a number of applications. So this is well explored where the core forming block is amorphous. And what I'm going to talk about is a twist on this, and it's actually a very simple twist. What we're going to look at is block copolymers where the core forming block, instead of being amorphous, it can actually crystallize. And remarkably, these types of structures have hardly been looked at in the literature. We stumbled into this area really by accident. And what we found is that if the core can crystallize, you get really profound changes in the self-assembly behavior and also what you can do with the resulting structures. Now, we actually got into this area by looking at block copolymers with a crystallizable core-forming block, which was based on one of the metal-containing polymer systems that we've developed in our group over the years. And this is the polymer system. It's called polyferrocenylsilane, or PFS. And this is the structure. So it's a metal-containing polymer. It contains metal and silicon. And these materials are made by ring-opening polymerization of these strained monomers. So these are very easy to make. We initially made them by simply taking these monomers and heating them, or using metal catalyst at room temperature, and you can make these polymers. It's a high molecular weight materials. If you look at the molecular weight distribution by gel permeation chromatography, you get a broad distribution. And the reason for this is these polymerizations, even though you get high molecular weight products, you get a range of chain lengths because there's spontaneous chain termination that occurs during the polymerization. So these monomers will open and then they start joining together, but you get a random termination step which can occur at any chain length. And so you get a range of chain lengths here, which you see in this distribution. And you can characterize this by the polydispersity index, or PDI. If all the chains were the same length, the value would be 1. And as you can see, it's a broad distribution here, and the value is 2.3. So you get uncontrolled chain termination. You also have an initiation step that's uncontrolled. In each of these processes, the initiation is a slow process, and so chains start to grow at different instants. And this will also lead to different chain lengths, which broaden the distribution. But you can get high molecular weight materials, as you can see here, which can be processed just like organic polymers. Now, over the years, we've invested a considerable effort in looking at the properties and applications of these materials. I'm not going to describe these in any detail at all in this lecture. This slide just shows some of the things you can do with these polymers. They're functional because of the presence of iron and silicon. So they act as excellent electron beam resists, so you can pattern them with electron beams. You can heat the patterns up and turn them into magnetic materials, so you can pattern the polymer and then heat it up to create iron nanoparticles, which in the presence of a hydrocarbon gas mixture will generate single-wall carbon nanotubes. And these are very well-defined carbon nanotubes that you can make by this method. And they've been used by other groups in devices, for example. Another interesting feature of these polymers is the ion center is a source of redox activity. You can go between ion 2 and ion 3 reversibly, just through oxidation and reduction, removal of an electron and adding an electron back. And this allows you to control the properties of these materials. For example, you can control their permeability. So if you make these materials into capsules, by oxidizing the capsule wall made of this material, you increase the permeability. And this is some work by groups in the Netherlands and Germany in showing that fluorescent dyes will permeate through the oxidized walls of these materials. So you can make redox active capsules. You can make gels, which as you oxidize and reduce them, they will swell and contract. And this has been used actually as a basis for photonic crystal display. So this display here is not an LED display. It's actually a photonic crystal display. It's made from a thin film of one of these gels on indium tin oxide, where the thin film is filled with silica microspheres. And when you oxidize the gel, it will expand and the spacing between the microspheres will change. And so the light that's scattered by Bragg scattering will change wavelength. And you can use this actually to make displays as shown here. And if you're interested in this, there's some papers down here which describe this. So these are interesting polymers in their own right. But in terms of this lecture, the only feature of them that is significant is the fact that if you have polymers with a very symmetrical structure, so if you have two methyl groups on silicon here, the polymer chains are regular enough in their structure to actually crystallize. And in the solid state, they will pack into crystallites. In fact, you can grow single crystals of this material. I'm going to refer to this as dimethyl PFS for the rest of the lecture. You can detect a melting transition. And you can show by X-ray diffraction that these are ordered crystalline structures. If you go from two methyl groups on silicon and just replace one of the methyls by another group, such as ethyl or phenyl, these structures now are not symmetric, and these can no longer pack into a crystalline lattice. And so these materials are completely amorphous. And you see this just going from the dimethyl material to the methyl ethyl. You go from a film-forming thermoplastic material into this amorphous gummy material here. With phenyl here, it's also amorphous. It's a glassy polymer, but there's no crystalline order here. So if you look by X-ray diffraction, you see no order for these materials, whereas this shows sharp peaks due to the scattering by the crystallites. So the block, in the block copolymers that I'm going to talk about, at least in the first part of the lecture, where the core forming block will crystallize, will be based on this, this block here with 2 methyl groups on silicon. So how does this polymer chain actually pack in the solid state? It's an interesting polymer chain. You've got ferrocene here with these 2 5-membered rings, an iron sandwich here, and you have this silicon spacer between the repeat units. To get some information on this, we made a model pentamer and crystallized it. It got a single crystal X-ray structure for this material. And the molecular structure has this transplanar zigzag conformation. The ferrocenes at the end of the structure are out of the plane of the rest of the molecule. But you'd expect it's the middle bit of the structure that's the model for the high polymer, which of course has a very high molecular weight. And if you look at how these pack in the solid state, you see this transplanar zigzag conformation in these planes here. So this is a top view, here's from the side. And if you imagine removing these end groups here and just continuing in this transplanar zigzag conformation for this dimethyl PFS analog here, we feel this is how the polymer chains, the analogous high polymer chains, will pack in the solid state. And if you compare the X-ray powder diffraction diagram pattern for this pentamer with the analogous crystallized high polymer, they're very, very similar, really supporting this type of conformation here. And this will become important later on. So to look at block copolymers, you've got to have a synthetic method that will allow you to make block copolymers. And the type of method you need is a living, what's called a living polymerization. And this is a polymerization where initiation is fast and there's no spontaneous chain termination or chain transfer, unlike the synthetic methods that I showed earlier for making PFS materials. And this can be achieved by actually a number of methods, but the method I'm going to describe is living anionic polymerization. So you treat this monomer with butyl lithium. It will ring open, the butyl group will attack silicon, butyl minus attacks silicon, you open this bond, and you generate polymer chains which have a lithiated chain end, and the butyl is at the other end of the chain. And this is a living polymerization because these polymer chains are stable in solution. They don't spontaneously terminate. And this allows you to add another monomer so that it will grow off the end. So you polymerize off the end, and you can make a block copolymer which you eventually terminate, but deliberately by adding for example, trimethylsilyl chloride, and this will give you a TMS group at the end. So this living route allows you to make block copolymers, and it also allows you a lot of control over the structure of this material. So this is a very controlled synthetic method. Now, to illustrate the living polymerization, because we're going to come back to this concept later in the lecture, if we just consider this first step here, this illustrates the living nature of the polymerization. And if you do this polymerization, you can take this living polymer you generate and deliberately terminate it by adding, for example, isopropanol, which will put a proton on the chain end. And if you do this, you can show a number of important features about this polymerization. The first is you can control the chain length, or the molecular weight of this polymer, simply by the ratio of the monomer to initiator. And the reason for this is, in a living polymerization, every initiator molecule generates one polymer chain. So the ratio of the initiator to the monomer will control the chain length here. So if this is 1 to 10, the value here will be 10. If it's 1 to 100, it should be 100. So as you vary this ratio, you can control the chain length. So this is one key feature of living polymerization. The other key feature is that the initiation step is fast. So the polymer chains all start to grow at the same instant, and they grow until the monomer is used up. And so the polymer chains you get all have essentially the same length. And if you look at the GPC trace, here's a GPC trace for the polymers that you can make using this method, and then you protonate them deliberately to isolate the polymer. And you get polydispersity indices very close to 1. And if you remember back to the GPC trace I showed earlier, which was really broad for the thermal and catalytic methods for making the polymers, where there's spontaneous chain termination, for example, you get a much broader molecular weight distribution. So you get very well-defined materials using this process. So you've got these two key features of a living polymerization. You can control chain length, and you get narrow length distributions. So this is one of the polymers that we're going to talk about. So here's the dimethyl PFS block, which can crystallize. And this is the polydimethyl siloxane block here in red. And this will form the corona or shell. We're going to put these materials in a solvent that is selective for the polysiloxane block. So hexanes will dissolve the polysiloxane block, but the PFS block is insoluble. If you use THF, both blocks are soluble. So if you want to dissolve this material to give individual block copolymer macromolecules that are dissolved in the medium, you can use THF. And there's two other polymers that I'm going to mention. There's the PFS polyisoprene. This is usually written the other way around, PI first and then PFS, because the synthetic method for making it involves the polymerization of isoprene first and then the ferrocenophane monomer. This gives you a polymer that has similar characteristics in terms of solubility to this one. So if you put this in hexanes, hexanes is a good solvent for the polyisoprene. It's a poor solvent for the crystallizable dimethyl PFS block. But a key feature of the polyisoprene is you have these vinyl groups present which allow you to do chemistry such as crosslinking, which we'll use later in the talk. And one other polymer I'll mention, this is PFS poly-2-vinyl pyridine. So here you have, again, a crystallizable dimethyl PFS core-forming block. The shell will be formed by poly-2-vinyl pyridine. This is a hydrophilic block, and so this is soluble in CHF, but also isopropanol, in which dimethyl PFS is insoluble. So this gives you a different type of polarity to the shell of the structure. So this is hydrophilic, whereas polyisoprene and polydimethyl siloxane are hydrophobic. Okay, so moving to self-assembly. So first what I wanted to show is what happens when you take a PFS block copolymer where we have an amorphous PFS block. So this is one with methyl ethyl or methyl phenyl PFS block and a polysiloxane co-block. Block ratio here is about 1:6. And if you put this into THF, THF is a good solvent for both blocks. So you get what are called unimers. So these are individually dispersed block copolymer macromolecules, because you're in a solvent that's good for both blocks. If you then add slowly hexanes, which is a selective solvent for polydimethyl siloxane, you'll get micelles where the PFS block will form the core. And in this case, with the case of methyl ethyl or methyl phenyl, you can look at the structures that you form in solution. You can do this by a number of methods. Here's transmission electron microscopy after solvent evaporation, and you get spherical micelles. And this is exactly what you'd expect based on the behavior of organic block copolymers where the core is amorphous. So what happens if you take exactly the same material, do exactly the same experiment, but instead of having the amorphous PFS block, you have one with two methyl groups on silicon that will crystallize, so dimethyl PFS. So here's the structure of this. It's identical, except you've got two methyl groups on the silicon for the core-forming dimethyl PFS block. You can put this in THF and then add hexanes. And what you find is, instead of getting spheres, you get a totally different morphology. You get cylinders. And the cylinders are of different length. You can see some short ones here and some very, very long ones. So there's no length control here. The lengths vary from about 500 to 2,000 nanometers. And in this process, what you can show is that the core of these micelles is crystalline. So here's a cartoon of these micelles. You've got the dimethyl PFS core in amber here, the polysiloxane in red. And you can show by X-ray diffraction and electron diffraction that this core is actually crystalline. And you can look at the spherical micelles that I showed in the previous slide where you had an amorphous PFS core forming block, and you can show that they're amorphous. So somehow the crystallization of the core forming block directs the self-assembly away from spheres to give you really well-defined cylinders, as you can see here. And the way we look at this process is that when you dissolve the block copolymer in THF and then add hexanes, the hexane starts to precipitate the PFS block, and at some point you induce crystallization, and so this is like a self-nucleation process or homogeneous nucleation. And then once you get crystals, this drives the self-assembly through to give you cylindrical structures. And the formation of the crystals can occur at any time. It can be quite slow, it can be quite rapid. And so these cylinders will start growing at different instants, and they grow until the block copolymer is used up. And so you get different lengths because they start growing at different times. So this is our thinking on this self-nucleation process. And you can do the same thing with these other materials. So if you take dimethyl PFS polyisoprene, dissolve in THF, add hexanes slowly, you generate, again, nice cylinders. Here with a polyisoprene corona in blue, again, you can show you have a crystalline dimethyl PFS core here. And you can do it with the hydrophobic analog, as hydrophilic analog, where you have a poly-2-vinyl pyridine shell here. Here you do the self-assembly in isopropanol, which is a good solvent for poly-2-vinyl pyridine. It's poor for the dimethyl PFS. But again, you get cylinders, crystalline dimethyl PFS core on a poly-2-vinyl pyridine corona, which stabilizes the structure and gives you colloidal stability in isopropanol here. An interesting thing to think about is how the PFS chains actually crystallize in the core here, and I'll come back to that issue a little bit later on. So these structures are stable in the selective solvent, and if you remove the solvent, they're still stable. So this is electron microscopy under vacuum, so these are still stable structures. They don't disintegrate when you remove the solvent. But of course, if you add a solvent that's good for both blocks, these will re-dissolve and go back to individual unimers. But you can actually make these structures permanently stable by introducing cross-linking into the shell of the structure. So if you take, for example, PFS polyisoprene cylinders here, so you can make in hexanes. If you add a good solvent for both blocks, such as THF, it will disassemble. But the polyisoprene chains have vinyl groups present, and you can actually join these together using platinum catalyzed hydrosilylation by adding a silane material. This will introduce covalent crosslinks, which I've indicated in red here, into the corona. And this fixes the structure in. This can no longer disassemble. So we write this as PFS block XL for cross-linked polyisoprene. And so this should now be stable in good solvents for both blocks. It should be blocked towards dispersion. And I'll show this in a moment. So you can do this irreversibly using platinum catalyzed hydrosilylation. But you can also just add platinum zero complexes, which will bind the olefinic groups. And then you make cross-links which are reversible. You can remove them again by adding a phosphine. So you can go back. So that you can actually do both irreversible cross-linking and reversible cross-linking. In this lecture, we'll mainly use irreversible cross-linking. So how is the stability of these structures? Well, if you take these materials now and put them in a solvent that's good for both blocks, you retain the structure. So these are now permanently stable, as you can see here. Once you've cross-linked them, they're much more stable structures. If you sonicate the uncross-linked micelles, they will decrease in size. You can show this by dynamic light scattering. But if you cross-link them, they don't change their size at all. So these are very stable. So if you have no cross-link, if you take long cylinders and sonicate them, they break up. And this is something we'll actually use very, very shortly in this lecture. Okay, so we can make these structures that have a crystalline core, and what was really surprising about them is the fact that you can actually, these cores are crystalline, sufficiently crystalline that you can do epitaxial growth off the ends of the cylinder. And this was actually discovered by Xiaosong Wang, who was postdoc in Toronto back in around 2005. So if you take one of these cylinders with a crystalline PFS core, and to illustrate this, we usually take a small cylinder, a short cylinder, which we make by sonication. I'll explain how we do this in a moment. If you take these structures, these we regard as seed structures. If you then add more unimer to them, for example, here we have PFS polyisoprene. If you add more unimers to this, the PFS blocks in the unimers you add will deposit, orient and add, and crystallize on the ends of the pre-existing seed micelle, and the structures will grow. And even more interestingly, if you do the same thing with a different corona-forming block, you will again deposit on the ends and grow the cylinder. In this case, you generate a segmented structure where you have different coronal chemistry along the structure. This is really a micelle analog of a block copolymer, so we call it a block co-micelle, where you have polydimethylsiloxane here in red from the added unimer at the ends, and the original seed is in the middle. So how do you do this? You take the seed micelles dispersed in a large amount of the selective solvent, such as hexanes. You add the unimers in a small amount of the common solvent, THF. The elongation appears to occur by epitaxial growth, by crystallization of the dimethyl PFS blocks on the ends of the pre-existing seed cylinders. Very, very importantly, this elongation process seems to be infinitely preferred to self-nucleation of the unimers to create new cylinders. You can imagine these unimers, when you add them to the seed micelle solution in the selective solvent, would actually form new cylinders. But this process is very slow. This is homogeneous nucleation process, whereas this seeded growth is kinetically much faster. And so we don't generate any new cylinders in this process. So all of the unimers you add to the pre-existing seeds. So in many ways, this is like a living covalent polymerization, which I illustrated earlier, for actually making the block copolymers in the first place. This is just on a totally different length scale. And you can make analogous materials. This is an analog of a block copolymer here that you'd make by living anionic polymerization, for example. Okay, so let me illustrate this process and what you can do with it. So to make the seeds, the way you can do this typically is to start with the cylinders you make by simple self-assembly by homogeneous nucleation. So you take the block copolymer, dissolve it in THF, which is a good solvent for both blocks. You add hexanes very slowly. This generates cylinders of different length because it's a self-nucleation process. The cylinders don't start growing at the same instant. So you get cylinders of different size. You can sonicate these down using a powerful sonicator to give you very small seeds, which you can see here, which will represent in this cartoon. You can then take these seeds and add more block copolymer in the form of unimers to the seeds, and the PFS block will then deposit on the ends of the seed, and the structure will grow. And if you carefully monitor this process, what you find is that you can control the length of the structures you generate. So based on the ratio of the unimer to the seed, this is the mass ratio. So when it's 5, you get cylinders that look like this. When it's 10, as you add more, they've grown. And here's 20, and here's 40. And so you can generate structures that all have the same length, and you can control the length. And here's the length distribution. You can create a polydispersity index in length here, and you see values all very close to 1. And you have control from 100 nanometers or so up to several microns in this process. If you plot the length against the ratio of the unimer to seed, you get a straight line, so you have length control. If you use this to generate, let's say you want cylinders of length 700, you can choose this ratio. You generate cylinders of length 700. If you look at the dispersity and length, it's a very narrow length distribution, you get a very narrow polydispersity index. So you get narrow length distribution. These are both characteristics of a living polymerization. So this is really like doing a living polymerization on a different length scale, and we called this process crystallization-driven living self-assembly because of the analogy with living covalent polymerizations. So it's a nice way for making controlled structures, but there's always, if you look at biology, it's always a good way to sober up. And biology has been doing this for millions and millions of years. Biology can make cylinders all of the same length. This is tobacco mosaic virus. It's made by assembling proteins around a strand of RNA. And these cylinders are all of length 300 and the width 18 nanometers. So biology can make structures where they're all the same length. But if you want to make tobacco mosaic virus with different lengths, if you don't want 300 nanometers, then that's a tough job. You've really got to do some synthetic biology to do this, and it's not a trivial matter. Whereas the block copolymer self-assembly method is crystallization-driven self-assembly is a very easy method to use to control the structure. So you can make structures all the same length, and this allows us to start looking at the properties of monodispersed samples of cylindrical micelles in solution. And you find that if you take a concentrated solution, they will tend to align, so they form pneumatic liquid crystals. And if you use an electric field, you can make them align really very well. And you can get very good order parameters where they will align with an electric field going across here. This is all done in this little cell here. And you can get alignments here that are very similar to the values you can get with tobacco mosaic virus. And this, when we noticed this got us excited because we wanted to find out the structure, the crystal structure, how the polymer chains pack in the core of the micelle. And this is a non-trivial business because if you have the micelles, if you isolate the micelles, they're always generally pointing in different directions. And to get the crystal structure of the core, you've got to really get them aligned. And this is a way of aligning them. So you can imagine probing the structure of these aligned structures by X-ray diffraction and then actually working out the crystal structure of the core and how the chains align. So to do this, we did this experiment at a synchrotron. And if you do this, you get really outstanding data. This is the small angle X-ray scattering that shows we've got very well-ordered pneumatic liquid crystal where the cylinders are all aligned in the same direction. And this is the X-ray diffraction of the core of the micelles. And you get a beautiful diffraction pattern. And this has allowed us to actually work out how the chains are packed. And they're actually packed perpendicular to the long axis of the cylinder. So if you do a cross-section, the polymer chains are in a trans-zigzag planar conformation across the cylinder like this. And the structure of the single crystal of the pensimer that I described earlier was vital in allowing us to actually determine this structure here. So you can make block structures. If you take seeds with one coronal block, so this is polyisoprene in blue, and add unimers with a different coronal block, we've got polydimethylsiloxane in red, you can make triblock comicelles. So this is a BAB triblock comicelle. And if you do careful electron microscopy, you could actually visualize the different segments in these structures. So the A block here is polyisoprene in the middle here, and B block, which looks thicker because the corona that contains silicon scatters electrons. You can see here, and you can do this this type of approach to build up really quite intricate structures. Here's an example where we start with seeds here, which are based on PFS poly-2-vinyl pyridine, with a little bit of a fluorescent tag at the end. So this is polyphenylene vinylene, just on the end of the coronal chain to give you a fluorescent tag. So that's the yellow seed we start with. We then add to that the block copolymer without the tag, so that adds each side. And then you add this one again, so it adds to the ends of that. And then you can build up structures like this, barcode type structures. And when you do this, there's always a plane of symmetry in the middle here because the seeds will grow from each side. So you always have a plane of symmetry with these structures. So this is a 9-block barcode structure. And then you can visualize these and also follow the whole process by confocal fluorescence microscopy. So these are the 5 blocks. This is the 9 block you have at the end. So you can really build up quite sophisticated structures. So how do you do this in the lab? It's very easy to do, and you can do it using wet solvents in air and in the fume hood. So initially what you do is you take the block copolymer and you dissolve it in a little bit of common solvent, THF, and then add hexanes, which is selective for the coronal block, and you leave it for 24 hours, and this is a slow process that forms cylinders by self-nucleation, and you get cylinders of different length. You then sonicate these down. You just put a powerful sonicator into the solution and break these down into small seeds. And then to the seeds, you add the unimer. This is the seeded growth process. This is a faster process than the self-nucleation. So all of the unimer that you add adds to the seeds. It doesn't form new cylinders. If it did, it would be a pretty useless process. So this is a key that the addition of the unimers to the seed is much faster than the formation of new micelles from the unimers. So this allows these structures to grow, and then you can add another unimers of another block copolymer on the end to make block structures. And so to illustrate the flexibility of this, here's some recent work we've done using PFS polysiloxane block copolymers, where we've attached different color fluorescent dyes to the coronal block. So you can make this material with a red dye here, or a green dye here, or a blue dye. And if you take each of these materials individually and self-assemble them, the one with the red dye, of course, will give micelles that's red, that fluoresce red. The green will fluoresce green, and the blue will fluoresce blue. But very interestingly, what you can also do is make analogues of random copolymers. So you can take mixtures of the material with a red dye, the material with the blue dye, the material with the green dye, in any ratio you want, and then control the structure of the resulting micelle. So you can even make white micelles by taking the right ratio of these block copolymers. You can go all over the chromaticity diagram and make any color you want within this structure just by varying the amounts of these that you use. So these are random co-micelles, if you like, that you generate here that generate the different colors according to the chromaticity diagram. Now, one problem in all the self-assembly, or the crystallization-driven self-assembly so far, is that we start with seeds which have 2 faces, so you grow from each end. So every structure you generate has a plane of symmetry in the middle. And so an interesting challenge that we're very interested in is trying to get structures that grow in one direction. Can we break the symmetry? This is a challenge. Biology can do it. Tubulin, for example, actually will actually grow in one direction to form microtubules. So biology can make structures self-assemble in one direction, but can we do it using crystallization-driven self-assembly? So what we want to do is make micelles where we can use crystallization-driven self-assembly and just grow in one direction. This will allow us, for example, to make AB block structures, which might be interesting, or ABC. And in fact, if you make one end hydrophobic, the other end hydrophilic, you might be able to self-assemble these into new types of structures, super micelles, which would be, up in terms of the length scale, length scales will be further up the length scale from normal micelles. So how do we do this? I'll share with you one method that we published last year. And the way we do this is we start off with tri-block co-micelles, where we have a PDMS We start off with a PDMS seed micelle with the red corona here, and then we grow on the end PFS PI, so we have a polyisoprene corona in blue on the ends. And what we then do is cross-link the polyisoprene using this platinum-catalyzed hydrosilylation. So this cross-links the ends. It doesn't cross-link the PDMS because it's got no vinyl groups. And the aim here is actually to block the ends of the cylinder through this cross-linking process. And our thinking here is that the ends of these cylinders, the polyisoprene chains, should spend some time in front of the faces here. And this is a reversible process unless you cross-link and fix it in to block the ends. So what we're hoping is we've blocked the ends of these structures. So that's the aim, and you can follow this by electron microscopy. So this is the TEM of the tri-block comicelles here. Once you cross-link the polyisoprene end coronas, you can see them, visualize them very easily in the electron microscopy. The next step is to dissolve out the middle, because this should be possible to solubilize since it's not cross-linked. You don't have a corona cross-linked central block. And the aim is to dissolve this out, and you can do this by adjusting the solvent and also heating a little bit. And if you can remove the middle, you should generate structures which have a newly exposed face here and here, which should be active to crystallization-driven self-assembly, whereas the other ends should be blocked. So again, you can monitor this process. Here's the tri-block comicelles. You dissolved out the middle. And here we have the ends, and we're hoping that one of these ends is active and the other end is blocked. So you take these and add more unimer to them, and what you find is they then grow in one direction. So if you add to these more PFS, PDMS, they will grow, and you can make AB block comicelles, as you can see here. And you can take these and now add PFS poly-2-vinyl pyridine, and that will give you an ABC structure. And this is interesting because it's an amphiphilic block comicelle, so the P2VP end in green is hydrophilic. The other two blocks are hydrophobic, so this end is hydrophobic. So if you put these in isopropanol, these should self-assemble through the PI and the PDMS end to give you core shell structures, which would be supermicelles. And this actually works very nicely. So you can make these supermicelle structures. Here's the core of the structure, and you can actually visualize them by optical microscopy. They're sufficiently large, and they're actually very stable structures. You can pick them up with optical tweezers. That's something we've recently been able to do. And you can actually visualize them by video microscopy. So here they are in a thin film of solution, and you can see them every so often floating across the slide here. So these are quite interesting structures, and we're actually doing quite a lot of work with these right now. So far, all of the crystallization-driven self-assembly I've described involves growth from the ends of cylinders. And first, I talked about growing from both ends, and just in that last part of the lecture, I've talked about how you can block one end and make it grow in one direction, which opens up new avenues for making asymmetric or non-centrosymmetric structures. But since this process just works on crystallization, you should be able to take any face of PFS that is crystalline and actually use this process. And this turns out to be the case. So you can take a homopolymer film. So this is dimethyl PFS homopolymer. You cast it as a film. You can anneal it to make it crystallize. You can then dip this in hexanes, add the block copolymer in the form of unimers, and this will initiate off of the crystallites in the surface of the crystallized film. And you can see here in B, this is early stages after adding the block copolymer. You can see cylinders starting to grow off of the surface, off of the steps in the surface. And in the end, the surface is completely covered with spaghetti. And you have this sort of micelle brush layer on the surface. So this is using a film. You can also use, instead of a homopolymer film, you can use nanocrystals of the homopolymer. So you can, if you take the homopolymer and dissolve it in THF and then squirt it into hexanes, it slowly precipitates. And in the early stages, you generate very, very small crystals of the homopolymer. And if you then add the block copolymer, you grow from the nanocrystals cylinders, and you can see them growing here. So as you add more block copolymer, they grow longer and longer, and you can make these star-like structures. So you can really make a lot of different structures using this approach. So we've been developing this approach over the last 5 or 6 years, and increasingly the thing we've been thinking about is what can you do with it? You can make nice structures, but what sort of applications can you think of for this type of process? And in finishing, I'll just show you a couple of examples of things we've recently done and recently been doing. You can functionalize these structures in principle 2 ways. You can use functionality on the corona or in the core. And to illustrate coronal functionalization, you can actually make well-defined nanowires from these structures. And the way we've done this is to make well-defined cylinders by crystallization-driven self-assembly of PFS poly-2-vinyl pyridine. And then we've templated the growth of conductive polyaniline in the emeraldine salt state around the cylinders so that you get well-defined nanowires. And these are the types of structures that you can make. These ones here of length 400 nanometers, width 15 nanometers. So you can vary the lengths here using crystallization-driven self-assembly. So we've made these of length between 200 and 1.1 microns. So this is just one example of the materials. Now, a key thing is that we're saying this is a nanowire. Does it actually conduct electricity? Is it conductive? And we've looked at this using a non-contact mode approach in collaboration with Andy Holton at Newcastle in the UK. And we use the method of scanning conductance microscopy, where you basically do, you look at, in tapping mode, you look at a phase image of the structure in the presence of a potential difference between the tip and the substrate. And if you have a conductive material, you see a change in the phase angle when you apply the DC bias. And so here's an illustration of some of the results. This is one of these emeraldine state coated nanowires looked at by scanning conductance microscopy. And a key plot here is shown here. This is the phase angle here. It shows the change as you apply the DC bias. And this is typical for a conductive material. So this shows that these structures are conductive. Unfortunately, this method, this non-contact method, doesn't give you an absolute value for the conductivity, but it tells you it's conductive, whereas the original micelles are insulating, which you can show by this method. So these structures really are conductive, so you can make well-defined nanowire type structures using this approach. Some other things we've been looking at in terms of functionalizing the corona is actually making encoded structures and nanopixels. So using these fluorescent dye functionalized block copolymers that I described earlier, instead of using all at the same time to make random structures, you can make block or segmented structures. So you can start with this is a micelle with no dyes in the corona. You can add the one with red dyes, which adds on each end. Then you add a bit of block copolymer with no dyes attached, and then you add the green one, and then another spacer layer with no dyes, and then the blue one, and you can build up these types of structures. So you can make these nanopixels, which you can visualize using confocal fluorescence microscopy. And these are ones of this type of structure. These are some of the ones where they've been grown in one direction here. So these are centrosymmetric, these are non-centrosymmetric. So these are quite interesting as encoded micelles. So for example, these 3 micelles look exactly the same by electron microscopy, but they're actually encoded with the colours of the Canadian, British and Irish flags here, just using the fluorescent dyes. So they're quite interesting materials. Finally, I'd like to talk about some recent work we've been doing. We're now working extensively in this area. And this is based on the idea that crystallization-driven self-assembly just relies on having a block that forms the core of the micelle that will crystallize. And there's many, many block copolymers that will crystallize. It doesn't have to be dimethyl PFS. We just stumbled into dimethyl PFS because we were studying the block copolymers in our lab. But you can actually think about doing exactly the same process with any block copolymer with a core forming block that will crystallize. And there's a number of groups around the world that have done really nice work with other crystallizable core forming blocks like polyethylene, the Schmalz group in Germany, for example. Polylactide block copolymers, work by Rachel O'Reilly and Andrew Dove at University of Warwick in the UK. So there's a lot of work being done on other materials. It's not limited in any way to PFS. And particular interest, at least from our standpoint, is looking at pi-conjugated polymers like polythiophene, which will crystallize. So you can make poly-3-hexylthiophene block PDMS. Poly-3-hexylthiophene is a crystallizable block, and so if you put this in a solvent that's good for the PDMS, you should get micelles where the P3HT will crystallize. First, you dissolve this in a solvent that's good for both blocks. So you put this into toluene. It gives you unimers. Toluene is good for both blocks. When you have the unimers, so when this is dissolved in solution, the poly-3-hexylthiophene repeat units, the thiophene rings are out of plane, okay, in solution because they're dissolved. And so the extent of pi conjugation is very low. So you have a yellow solution. But then as you add a solvent that's selective for PDMS, such as ether or heptane or hexane, this will assemble. The poly-3-hexylthiophene will precipitate and crystallize, and you get micelles. As they crystallize, you enforce coplanarity of the thiophene units, and this gives you extended pi conjugation, and so you see a red shift in the color, and it goes purple. And you can show by electron microscopy you have these fiber-like cylindrical structures, and you can sonicate these down to give seeds. And then you can think about growing the structures. If you add block copolymer unimers to the seeds, they will grow, and they do grow. But there's a problem, and that is they grow up to about 200 nanometers and then stop growing. And a problem here, we think, is the fact that the poly-3-hexylthiophene crystallizes so fast you introduce defects into the core, and eventually it will no longer grow because there's just so many defects in the core of the micelle. And we've been exploring ways to get around this. Here's another system. This is poly-3-hexylthiophene poly-2-vinyl pyridine. Again, you can make seeds and then you can grow these structures. In this case, you can grow them a bit longer, up to about 300 nanometers, but you have the same problem where we believe you're introducing defects. So as you try and grow longer and longer, you find their real length decreases relative to the length you expect. So we've been trying to solve this problem. We've recently had success in that. We can slow down the self-assembly and make much better cores, and we can get longer structures up to about 700 to 800 nanometers. If you look at the cylinders you generate, you can show that you have extended pi conjugation in the core of the micelle. So if we're going to look at charge transport, electronic properties, think of these as nanowires, for example. You need to have a high order in the core. And when you go from, if you look at the UV vis, as you go from unimers to micelles, you get this red shift. And there's this telltale shoulder here, which is indicative of a crystalline core for the poly-3-hexylthiophene. This is well known in the literature. And you get this shift as you get extended pi conjugation. And as you go from unimer to the micelles, you get fluorescence quenching, and this is also what you'd expect for these structures. So very recently, we've been able to make the first examples of block co-micelles from poly-3-hexylthiophene. We started with a seed. This is the seed we started with, which is P3HT-PTVP. You can add to that P3HT polyethylene oxide, so this grows off the end, and then you can add more P3HT-PTVP so that you can make pentablocks. So you can make block structures, but what you find is there's defects. Here's an unreacted seed. For some reason it didn't work. So we've still got work to do to really perfect this process. But we are making progress, and one of the things we're really trying to target is structures where we have pi-conjugated cores with different band gaps. So you have a heterojunction in the core of these structures, and these are things we're really working hard at the moment, and hopefully we'll be able to report these in the next, hopefully in the next year. Okay, so let me just summarize. I've talked about crystallization-driven living self-assembly of block copolymers, which is an interesting new approach to making structures on this length scale of about 10 to 2,000 nanometers. So you can make monodisperse, soft matter-based materials. You can spatially control charge and polarity through segmentation of the structures. There's a lot of people working on very nice methods which are fully complementary to what I've talked about. But each method has advantages and disadvantages. There's other methods which are more what you might call step growth methods, where people take nanorods, functionalize the ends, and then can join them together. There's work by Kumacheva. There's other related work by Müller, Mullen, Schenning, and Meyer. So this is an active area where there's lots of, I think, interesting methods, synthetic methods being developed. For crystallization-driven self-assembly, there's analogies with living covalent polymerizations, and also with the formation of viruses and protein fibers in biology. All of these are cooperative nucleated growth or nucleation elongation processes. So this is, in many ways, a synthetic analog of living covalent polymerizations, and also biological processes such as virus formation and protein fiber formation. They're all conceptually related. And you can make all sorts of structures, block co-micelles, you can make non-centrosymmetric structures, you can make multi-arm structures. All you need is a crystalline core forming block, and we discovered this initially with polyferrocenylsilane. You can do it with many other materials, polyethylenes, I talked about polythiophene, but there's work on polylactides, polycaprolactone, polyacrylonitrile, and I think many others. There's many people working on other materials, and potential applications I've mentioned in the lecture. I'm really privileged to work with a fantastic group of highly motivated, talented, and very nice people. Here's, in this slide, these are the people who did most of the work that I described, certainly the new work that I described in this lecture. Joe Gilroy was a Marie Curie fellow in my group. He's now assistant professor at Western Ontario. Huibin Chu is a Marie Curie fellow, a postdoc in my group, and he's now looking for jobs in China. He's just got an interview in China, academic job. Sanjeev Patra was another Marie Curie fellow-supported, fellowship-supported postdoc. He's professor at the Indian Institute of Technology now in Kharagpur. Nina McGrath was a PhD student working on our group. She did the work on the polyaniline-coated micelles. She's now working in industry in mainland Europe. Zach Hudson has got a Marie Curie fellowship. He's working in my group. He did work on the fluorescent micelles. Paul Rupar, another Marie Curie. These are Canadian. Joe Gilroy's Canadian. Zach Hudson's Canadian. Paul is now a faculty member at the University of Alabama in the States. Lawrence Shaban was a PhD student with me. He's now postdocing with Marcus Antionetti in Berlin. George Whittle is my senior research associate. He does a fantastic job on all the projects in our group, and he's intimately involved in all the things I described. Jess Guider has done great work on the polythiophene systems I described. She's still in our group as a postdoc. And Dave Lunn is a PhD student who's worked on the fluorescent micelles with Zach and many other projects. I'd like to also thank Xiaosong Wang. He really started the work on crystallization-driven self-assembly when he was with us, and it's been great to see him here at Waterloo, and other people involved, Rumen Ahmed, Felix Shaka, Emily Canaston, Torben Gett, Graham Cambridge, Jishu Kwan, Fong Hei, and Gerald Guerin. We have a great collaboration with Mitch Winnick on all of the work that I've described, or virtually all of it. So all the work on crystallization-driven self-assembly is in collaboration with Mitch at Toronto, and in fact before Christmas I met with Mitch and his current group members that are working on this project, and we've still got a lot of collaborative work going on. Rob Richardson in physics at Bristol for the scattering work, Steve Mann, Charles Full, John Mitchells at Bristol, Andy Holton and Ben Horrocks for the scanning conductance microscopy. Funding from these places, and a number of people have asked me where Bristol is. It's right over the other side of the country here, which is 2 hours drive in the UK. It's not quite as big as Canada. And this is the synthetic chemistry building, and we're located on the 4th floor here in Bristol. And finally, I'd like to thank you for your very kind attention. Thank you. We haven't done that. I think it's a really nice idea because you might be able to generate helical structures and so on. We haven't really looked at that. I think it's an open area. There is a paper from Todd Emmerich's group at UMass where they looked at the self-assembly, crystallization-driven self-assembly of a polythiophene block copolymer and reported the formation of helices, but it wasn't really explained. They had a polyethylene oxide corona block, and they added metal ions, which presumably complex to the PEO. But it's not really explained how they see the structures that they do see, but it's pretty convincing that they form helices. But I think this is a very interesting area. We haven't done anything on that yet. Yeah. That's probably you can, yeah. The reason you get cylinders in all, I didn't really go into detail on this, but the ones that we get cylinders where the coronal block is much longer than the core forming block. And so the formation of a cylinder is really a consequence of competition of the crystallization and the coronal chains that repel one another. And if you have a long coronal block, it tends to favor some degree of curvature, which of course if you have a platelet structure you don't have. Once you reduce the length of the coronal block, then you get platelet structures. And we can do that. And we've recently been working on growing those in a controlled way as well. And it basically works. So you can do it with you can do it in 2 dimensions as well. Yeah. These systems are not really equilibrium systems. They're really kinetically trapped. Okay, there's no unimer exchange. So you can take mixtures of cylinders of different length and mix them up, and if you leave them, you end up with the same mixture. They don't exchange and all equilibrate, so you get some length that's in between the 2 initial components. So they're kinetically trapped systems. So concepts such as critical micelle concentration don't really apply to these systems. There's no unimer left at all, and the lengths of the cylinders that you get in self-nucleation seems to be related to the number of crystalline seeds that you generate through the self-nucleation process, which is very difficult to control, but there is some control. For example, if you add if you make the micelles in a solution where there's a small amount of common solvent, you still get cylinders, but you get much longer cylinders. So presumably you generate a much lower concentration of crystallite seeds that initiate the formation of the cylinders under those conditions, which kind of makes sense if there's some common solvent present. But there's a hell of a lot we don't know about these systems. Even very basic studies of many of these systems are sorely needed, I think. But they're quite different from the sort of equilibrium systems that you often see, well, certainly with surfactants. With block copolymers, they're often non-equilibrium. But these are, as far as we can tell, are completely kinetically trapped, at least at room temperature. If you heat them up, then you can get them to dissociate, and then they behave like, you can get equilibrium under those conditions. Yeah, this is a really interesting question. This does not happen as far as we can tell. And we've left solution of the micelles for a year, and you don't see any evidence of that. So presumably at the end of the micelles, there's coronal chains that sort of push outwards, and they sterically stabilize the structures. And also the structures, to join them together, you need some sort of glue. And we have actually, I don't know if I've got, hopefully I've got an example of that, which I can illustrate in here. So yeah, here we go. We've actually been able to join structures together, and the way you do it is you can add some homopolymer. So if you take well-defined seeds, and these are, as far as we can tell, indefinitely stable. They don't join together. But if you add some homopolymer, this should add to the end, and then there's no steric stabilization, so you'd expect the structures to come together, and that's what you find. And you can start growing these structures together, and we've been able to control it to some extent. Here's somewhere we've been able to glue these together, so you can actually join them using homopolymer or block copolymer, where you have a very short coronal block. Use these ones, and you can really start to actually control it quite nicely. But if you don't do that, you have to do it deliberately. Otherwise, they're as far as we can tell, indefinitely stable. Yeah, no, there's no energy transfer process. We put in a spacer layer to prevent energy transfer between the the dyes can be. You excite the dyes and then view them. You can view the red, the green, and the blue under under different conditions under the confocal fluorescent microscope. So
you have to broadly excite them.
Yeah, that's how it's done. Yeah, the systems were designed so that you didn't get crosstalk between the dyes. Yeah. Okay, so this is a good question, because in solution they don't do this. You can show by light scattering that they appear to be well dispersed in solution. Let's take an example. I think this one probably is good. So this is this is after solvent evaporation. So when you remove the solvent, the micelles have a choice. They can either interact with the surface or they can interact with each other, and they prefer to, I think, interact with each other. So here they're interacting through the coronal blocks, and so this bundling is the TEM. I didn't mention this. The TEM only shows the core of the micelles, because the core contains iron, so it scatters much more strongly. So each of these, the corona is kind of invisible here, but that's what actually spaces out these structures. And they prefer to interact with one another through the corona than interact with the surface. So that's why they bundle. So in solution they don't bundle, but after solvent evaporation they tend to.
Just to follow up on that, there is no any directions.
Yeah, these are certainly more. These appear more rigid than cylindrical micelles we have in an amorphous core, and you can see they're kind of straighter.
But this would be for the soluble part, right?
The soluble part, yeah.
Meaning that the crystalline core induces rigidity to the.
Yeah. I don't know if we've got it. I mean, this is a really interesting point. I don't think we have any evidence for that. We never really thought about that, but I think it's a really interesting point. I don't know what influence the crystalline core actually has on the corona, but it may not be so significant, because I draw these with I've talked about the core being crystalline, and but what happens at the surface of the core? Okay, they seem to be cylindrical, and if it's crystalline, it's got to be rectangular, presumably the core. So we think probably at the edge of the core there's disorder, and you have an amorphous region around the outside, which is probably where the chain folding takes place, because we know that in these cores, we know that how the chains pack, and the diameter of the core is about a third of the extended chain length of the core forming block. So there's clearly chain folds. And so there's got to be an amorphous region around the outside. So actually where the coronal chains are attached, almost certainly it's in an amorphous region. So it may not have an effect, but I think it's a really interesting point, and it may be possible to control things if you can make rectangular structures. With platelets it might be relevant, for example.
Have you looked at other functional groups on the corona that would enable like other mechanisms for temporary or permanent cross-linking? So you show hydrosylation or flattened chelation.
Yeah.
But there's got to be...
There's loads you can use, yeah. The challenge is developing one that works in the selective solvent. And in the case, as I described there, we're dealing with hexanes. So platinum catalyzed hydrosylation is very nice because the two components are both hexane soluble. Carsed's catalyst you can buy.
But click chemistry, photocycle additions.
Yeah, you can do any of those. Yeah. And we've done many of those. We have a whole suite of different methods that will work. And the way we attach these fluorescent dyes is using thiolene chemistry, and you can adapt that. If you use a difunctional component for that system, then you'll get cross-linking. So you can do it. It's very flexible. You just have to think about the main thing to think about is the solvent system, and making sure you don't get precipitation or anything. But yeah, you can functionalize. We put a whole variety of things on there. You can put phosphine groups on there and then use metals, for example. There's a whole load of work that we've done, which I didn't talk about. But yeah, it's very flexible.